Now, even though the eukaryotic genome is mostly made of non-coding DNA, there's still a bunch of different types of protein encoding genes. First and foremost are single copy genes, which is just a single copy of a unique gene in the genome. And this is probably how you pictured most genes being in the genome. But actually, some genes are tandem clusters, and these are clusters of identical copies of genes that are actually transcribed simultaneously. And this is in part to help increase the output of transcription of these genes. So these tend to be genes that are needed in high demand all at once. Now you can see here in this picture that there are a ton of different types of proteins that our genes encode for. And among those are things we've seen like receptors and structural proteins and cell junction proteins, chaperones to help with folding. There's a whole mess of different types of proteins that our genes encode for. Now, how do tandem clusters form? Well, they form through a process called gene duplication, where an extra copy of a gene is added to the chromosome, and this can be due to something called unequal crossover. An unequal crossover is a misalignment of the chromatids during crossing over. So if you look at our example right here, we have a crossover that's going to occur, but oops, they're swapping this sequence out over here. Uh-oh, that's going to lead to unequal crossover, which you see right here. Boom. Now we have duplicated that B gene. So that is how gene duplication can lead to tandem clusters in addition to other things. So let's turn the page and take a look at some other examples.